Oh, hi everybody. You caught me here in the A&P lab hanging out with all my friends. Is that sad? I don't think so. I'm enough of an A&P nerd that I actually don't think so. So, here we are in the second semester, Bio 224, Human Anatomy and Physiology 2, for lab number 1, which is a cardiovascular lab, like a few of the first ones are if you've looked ahead. This one is all about the heart. So have at the ready either a digital or a hard copy of lab number 1, your student lab manual, free to you in this same canvas shell. Get it out, take a look at it, and let me give you some free advice for what many people find one of the more challenging things in lab number 1. Now, as you know, because we've already gone through the list of things to know for this lab, the big chunk of the lab is the heart models and the coronary blood vessels. A little bit of sheep hearts. ECG will be very straightforward for you. Heart sounds, not much to that. But those heart models with the coronary vessels can give you a lot of trouble if you're not careful. So let's be careful. So get ready, buckle up. It's going to be a fun ride. Now, I know what you're thinking. Hey, with this online stuff, I was supposed to not have to sit in front of somebody and watch them talk at a whiteboard. But au contraire, you do. At least a little bit in 224 lab. So have those two pictures ready, coronary arteries, coronary veins. Also, have at the ready your handy-dandy lab manual opened to page number 6, which is where I am. And I want you to look at the right-hand side of page 6, about 2/3 of the way down where it says cardiac structures. And I want you to look at 3 things that are listed under that heading. The anterior interventricular sulcus, the coronary sulcus or AV groove, and the posterior interventricular sulcus. Find those 3 things, and I know you're saying, this is just start a lab, have some pity on us, have a heart. Well, I have a heart. Actually, I have 2 hearts, this one and this one. So, I am having a heart. Let's talk about these structures. So a sulcus, you might recall from your first semester class, is a depression, a groove, a valley in something. Remember the sulci or sulcuses in the nervous system that you probably studied and probably remember. The heart has 3 major sulci in it, sulcuses in it. So I would like you to watch where my finger is right here on the front side of the heart at a slight angle. There's a groove in this heart model, a valley called the anterior interventricular sulcus, a groove between the 2 ventricles. So right here where my finger's running, where these blood vessels live is called the anterior interventricular sulcus. On the back side, where these 2 coronary vessels live, is the posterior interventricular sulcus. And if I was to grab this heart model like I was going to strangle it, like this, the place where my fingers go right here is the coronary sulcus. The coronary sulcus or the AV groove. So 3 sulcuses or depressions on this heart, the coronary sulcus right here, the anterior interventricular sulcus, and the posterior interventricular sulcus. Now, why do I take the time to point these things out to you? Because these are valleys on the surface of the heart. And what do we often find in the bottom of a valley? A river or a stream. In this case, coronary blood vessels. So I would like you to look at those two pictures that you have in your textbook, one of arteries, one of veins, and I would like you to do this for me. Tell me what is the name of the artery, the coronary artery, that runs down the middle of the anterior interventricular sulcus. I'll wait. Don't look at me, look at the page. Seriously, look at the page. What is the name of the artery that runs down the anterior interventricular sulcus? Well, I bet you got it. It's the anterior interventricular artery. So I would like you to watch what I'm going to do here, because I didn't spend all that money at the office supply store for nothing. I got all these colored markers, and they're just sitting here drying out while we're doing online learning, so I'm going to go ahead and use them. And what I will do, and I suggest you do as well, draw yourself essentially a heart, an anterior view, and then right next to it, a posterior view. And guess what? I'm going to use red and blue markers. I've got red, I've got blue, and I'm going to draw in these coronary arteries and veins. I suggest you do the same thing. Get the crayons, get the markers, get the highlighters, make the picture. This is what we call, think back to 223, procedural memory. You will remember it better if you actually do it. Don't just kick back and watch me do it like you're streaming something on Netflix or Amazon or Hulu. Oh yeah, it's cool, make some popcorn, have a good time, but take some action. Save yourself the time and effort in memorizing these structures by doing something, a physical action. Drawing these pictures will literally help you learn them faster. So again, the blood vessel that runs down the front of the heart in the anterior interventricular sulcus is the what? Anterior interventricular artery. Go ahead and label it in your picture. Using the same two pictures in your textbook, what is the name of the blue coronary vein that's paired up with this one on the anterior surface of the heart? What's the name of the coronary vein that lies right next to the anterior interventricular artery? What's the blue one? The great cardiac vein. They're a pair. They're a couple. They live together at the bottom of this sulcus. So it doesn't really matter what model you see. They all look slightly different, but the red artery is the anterior interventricular artery. The blue vein is what? The great cardiac vein. Look at the back side of the heart in your diagram. These are the things they have shaded slightly differently for you to look at, so look at those. What are the two vessels, artery and vein, that live in the posterior interventricular sulcus. Red one, blue one. See the pair. The red one is called the what? Yes, you got it. The posterior interventricular artery. I'll say that again. The posterior interventricular artery. And what's the blue one that lives next to it? The middle cardiac vein. Notice I didn't label mine. You have to do something. So label yours. Yes, make the picture labeled. So on the front side or anterior surface in this sulcus, what do I have? Anterior interventricular artery, great cardiac vein. On the back side, living in that posterior interventricular sulcus, what do I have? Posterior interventricular artery, middle cardiac vein. I also have some vessels living in the coronary sulcus, a few of which we need to know the names of. So I would like you to start over here with me. And what coronary artery leads into the anterior interventricular artery, then branches and goes around back in the coronary sulcus? Look at the picture. Take a moment. We've got time. This one is the what? You see it here in the picture. The left coronary artery, which branches into the anterior interventricular artery and the, pause me until you figure it out. It goes in a circle. Pause me. The circumflex artery. Again, I'm not labeling them here in my picture. I already know them. You're the one who has to learn them. So make sure you're labeling these things. Left coronary artery, anterior interventricular artery, circumflex artery. See that? Do you see what I'm doing here? I'm creating diagrams showing me these coronary vessels. What do I have over here? I have a right coronary artery. So it's always his left and right. So, I have a left coronary artery, anterior interventricular, circumflex. I have a right coronary artery, which goes all the way over here and then shoots down the side as the what? Right marginal artery. Again, you need to be labeling all these. Not me, you. Left coronary artery, right coronary artery. Circumflex artery, anterior interventricular artery, right marginal artery. Posterior interventricular artery, which actually just comes from this guy wrapping all the way around here like that. What else is there to know? Well, don't I have, look at the other side, the venous picture, don't I also have a blue blood vessel coming down this side that I need to know the name of? What's that one called? The small cardiac vein. These two are a pair. The right marginal artery, sometimes called the right marginal branch of the right coronary artery, the right marginal branch, and the small cardiac vein. What's on the front? Great cardiac vein. What's on the back? Middle cardiac vein. What other blood vessel is on the back side of this heart that I have to know? A great big sort of bulgy thing like this in the coronary sulcus called the coronary sinus. That's the coronary sinus. Now let me show you some of these on this particular model. In fact, I think I'll just wait and show them to you a little closer up. But label these blood vessels, all of the coronary vessels that you need to know, and think of them as pairs. Posterior interventricular and middle cardiac. Right marginal artery, small cardiac. Anterior interventricular, great cardiac vein. They're paired up in those sulcuses. Draw these things, label these things. Don't just sit back passively and watch me do it. Okay, now that you've hopefully drawn a few coronary vessels on the surface, anterior, posterior, and lateral surfaces of a heart, let's take a look at some of these actual heart models that we have in lab, or to be more direct with you, heart models that I could ask you questions about on your quiz or practical exam. So notice first that I have 5 different heart models in this lab, any one of which I could ask you questions about for your first quiz or practical. Now certainly if you look closely, you probably see that the heart model on your far right and the one second from the left are the same exact model, just 2 different sizes, so that makes it kind of easy. But since no model is perfect, oh yes, I know you might be perfect, but my cardiac models are not perfect. Not every model shows everything equally well, so I have to take you through all 5. So have at the ready your student lab manual, and what I think I will do is just take you through different sections for each of these 5 models. So let's start over here. We're on page 6 of Lab 1, layers of the heart wall and the heart chambers. Let's do those 7 anatomical things first on each one of these models. So let's go with our most generous heart model, this jumbo one, very, very large, and let's talk about the 3 cardiac layers, the 3 layers of the heart wall. So, I'm sorry, I'm a heartbreaker here, got to break the heart, yes, haha. So, there we go. Let's take a look at these 3 layers of the cardiac wall. I always recommend that you begin with the myocardium, which of course is more than 90%, about 95% of the cardiac wall is myocardium right here. So this is the muscle, myo, M-Y-O, meaning the muscle. So the myocardium is this thick layer right here that you see composed of those cardiac muscle cells you learn about and talk about in lecture. The epicardium, out here where my finger is, let me use my pointer, the most superficial layer of the heart wall, it's the epicardium that actually uses a little bit of connective tissue to hold these coronary vessels down onto the surface of the heart. So epicardium, out here, most superficial, myocardium, and then the endocardium, the internal layer, I can see here looking a little bit shiny. It's where I have lots of what we call endocardial modifications, the papillary muscles and the cords and the valves and so on. So, epicardium, myocardium, endocardium. They would be easy to indicate on a model this thick, and I always like to use the left ventricle because it's so thick as compared to the right ventricular wall over here, not quite so thick. Myocardium, endocardium, epicardium. And this heart, like all of our models, has 4 chambers. 2 atria, 2 ventricles. So if I dismantle it a little bit, I can see in the bottom, right here, shaped like a V. Hey, that's a handy little memory trick if you've never learned the heart chambers before. Shaped like a V, the 2 ventricles. Now remember, this person is facing you. So here is the left ventricle. Here is the right ventricle. Notice on this model, I already mentioned it, and it'll really help you out with the sheep heart dissection. The left lateral ventricular wall is always much thicker than the right ventricular wall. It's a way to orient yourself whenever you see a heart model or a heart dissection. So left ventricle, right ventricle. The other 2 chambers are the atria. On this model, I sort of have to angle it a little bit for you to see them. Left atrium up here, right atrium over here. The atria on this particular heart model are not that great or easy to see, but I'll show them to you one more time. So I have the right atrium right there, left atrium right there. You'll see how it's a little bit better on some of my other models. Let's look at this one. So if we take a look at this model and I open it up, I can see very nicely the 3 layers: epicardium, myocardium, endocardium. Let's go over here. Epicardium, myocardium, where my probe is right now, endocardium, the inside or innermost layer, which is just some simple squamous epithelium for you 223 heads. The chambers, remember he's facing you, facing the camera. Left ventricle, look at that thicker myocardium. Left ventricle, right ventricle, left atrium, right atrium. 4 chambers. You can see all 4 of them in this model pretty nicely, I think. Left and right ventricles, left and right atria. Next model, sort of an old-fashioned looking model with these neat little hinged doors. Notice this. So let's do the same thing with this one. Epicardium, out here I'm tapping, myocardium, shiny squamous endocardium. Epi, myo, endocardium. Left ventricle, right ventricle, left atrium, right atrium. Notice how the ventricles are shaped like V's. And I always say that the atria are shaped sort of like a lowercase a. You have to use your imagination to see it a little bit, but I think you can. Now let's get another heart model. This one's very nice heart model. Opens up just like the others. So here we have a very nice depiction of the cardiac wall. Let's zoom in a little bit for you. And I'll zoom in more in the practice section of this online lab. Have no fear, I'll do my best to zoom in so you can see these things pretty distinctly. But Notice right here where my pointer is the epicardium, myocardium, endocardium. Epicardium, myocardium, endocardium. Very nice on this particular model for us to look at. The left ventricular myocardium on the lateral side is not quite as thick here as I would like it to be, but then the model builder didn't ask me for my personal opinion when he or she constructed the model. But I can definitely see these three layers of the cardiac wall very nicely. Left ventricle, right ventricle, right atrium, right here, and the left atrium on this particular model is a little tougher to see. It's back here, and it doesn't open up the best in the world, but it does. There's the left atrium right there in this one. So the four chambers, left and right ventricle, right atrium, right atrium, left atrium. Now let's look at our last cardiac model, which is the same as our second cardiac model. Here it is right here. So we can do exactly the same sort of thing if I open it up. So let's look at the layers of the cardiac wall. Epicardium, myocardium, shiny endocardium in there. Let's go to the right ventricle, epicardium, myocardium, see the color change right here on this model, endocardium, right there. Now I know it seems kind of silly that I keep repeating this over and over again, but this is what you would do if you were here and you were smart, and you are all smart. So I'm just doing what you would do if you were here. Let's look at the chambers, which I can see quite nicely on this particular model. The left ventricle, notice the thicker myocardium right here. Right ventricle, right atrium, and I have to go way back here, pop off this rearward section, the left atrium right there. 4 chambers. We humans have a 4-chambered 2-system cardiovascular system. So we are right atrium and ventricle, left atrium and ventricle, like that. So you've seen it on all 5 models, the layers of the cardiac wall and the 4 chambers. And of course, I will try to give you some good video practice in the practice section. Okay, next up, let's look at the great vessels and the heart valves on each model. The great vessels and the heart valve. So I'm still on page 6 of lab number 1, and we'll look at each model in turn that is potentially fair game for you on a quiz or a practical. And let's start with jumbo here. So as you have probably learned or will shortly learn in your lecture, the great vessels are those blood vessels that are actually attached to one of the heart's 4 chambers. So remember, we had 4 chambers, 2 ventricles, 2 atria, and each one has a great vessel or vessels attached to it. either carrying blood out of this particular chamber or into this particular chamber, it will very much help you. See my wagging finger here. Very much help you in lecture when you memorize the pathway of blood through the heart. So that's part of your lecture professor, even if that's me, that's part of their job, is to go through that with you, or at least assign that to you. How's that? But it'll help you a lot when you know the great vessels as you see them here on these models. Now you will notice some coloration here that's a little different on this jumbo model. Sort of this purplish color and then standard red and blue. Generally speaking, red is for artery, blue is for veins, in general. If there's a goofy blood vessel, meaning one that's not standard, sometimes it gets a non-red, non-blue color like this one. Red for arteries, blue for veins. Now be aware, you do not have red arteries and blue veins. Your blood vessels, if you were to actually see these two, are not red and blue like this. They're just painted to help us identify them one versus the other. And the same will be true next week when we're doing peripheral blood vessels. But for now, for the most part, just keep in mind that red is for arteries, blue is for veins, and off colors are for veins or arteries that are a little different. Let's begin with this great big giant artery that we see right here in red. This is the aorta. The aorta, which is connected to, exits your left ventricle. Right up there, if you can see where my marker goes, right up there is where this thing exits the left ventricle. This giant artery comes out and makes a huge U-turn that I see right here, which is the aortic arch. So the aorta coming out of the left ventricle makes a big arch. You'll see that a little more pronounced on a few of the other models. So the first great vessel connected to the left ventricle is the aorta. Comes out, makes a big arch. Then I have what's called the pulmonary trunk. This one you always see as most anterior on these heart models, and it's this sort of purplish one that comes out and goes at an angle. It almost always exhibits a sharp angle here toward the patient's left. If I use an Arnie Allner word, my dad, this is kittywampus. This comes across the surface of the heart going kittywampus or diagonally. This thing is the pulmonary trunk, which exits the right ventricle. So if you follow my finger, it now goes into this pulmonary trunk. Now, why the odd color? This thing is actually an artery because it's carrying blood away from the heart. But since, as you will talk about in your lecture, it carries deoxygenated blood, it is often given a blue color, but this particular model maker wanted to show us, you know, it's a little goofy, an artery that's not red, so they made it sort of an off blue color. So the pulmonary trunk, right here, which very quickly branches into pulmonary arteries. I can see this right back here, everybody. So the pulmonary trunk branches. This is carrying blood to my two lungs. The term pulmonary, of course, refers to lung. So pulmonary trunk, right here where my finger is, branching into the pulmonary arteries, which this model cuts off pretty quickly here and here. You can see those. So again, just to review, the aorta is attached to the left ventricle, the pulmonary trunk is attached to the right ventricle. But the two atria up here have their own great vessels, which are somewhat difficult to see on this particular model, to see that they're connected to the chambers. So we have pulmonary veins and the superior and inferior vena cavas. Now let me show them to you on this model after I give you a little explanation. The pulmonary trunk and then pulmonary arteries carry blood to my two lungs. Pulmonary arteries to the lungs. Pulmonary veins back from the lungs. Since the blood has gone to my lungs, done the hokey pokey with CO2 and O2, you learn all about in our respiratory chapter, it comes back as oxygenated blood, so these are usually made red. Pulmonary veins. So contrary to the normal coloring pattern, these are two veins that are red, not blue. Typically coming in as 2 and 2 like this, dumping blood into the left atrium. Again, I'm not really crazy about the atria on this model. It'll be a little clearer on the others. So the pulmonary veins carrying blood into the left atrium. This model, however, has a perfectly good superior vena cava carrying blood down from your head, neck, shoulders, arms into the right atrium. And we have an inferior vena cava right here, this blue one, carrying blood into the right atrium. So my finger is in the right atrium right now. The inferior vena cava carrying blood from the lower 2/3, 3/4 of my body like that into the right atrium. Superior, inferior vena cava. Those blood vessels actually connected to one of the heart's 4 chambers are called the great vessels. We're going to look at those on each model. Now let's talk about the valves. Your heart, like it has 4 chambers, has 4 valves. And the job of the valves is to prevent blood from moving backwards. So what I have are 2 very large valves here and here, separating the 2 atria from the 2 ventricles. I'll say that again. 2 very large, serious looking valves here and here, separating the atria and the ventricles. These are often called the atrioventricular valves because they separate an atrium and a ventricle, but much more common than this, you will hear them referred to by how many cusps they have. Bicuspid over here on the left side, this is the bicuspid valve, and the tricuspid valve over here on the right side, tricuspid valve. Now, in the medical world, if your future client, customer, patient is a Homo sapien, you might as well get in the habit of saying mitral valve and tricuspid valve. Mitral and tricuspid. Either name here is fine for your quiz or your practical. You could say bicuspid or mitral. Now, this name actually comes from the fact that this valve has 2 cups that close together. Actually, it'd be upside down like that, but like the hat on a bishop, a chess piece, or the hat, you know, some of you, maybe you watched that show, Schitt's Creek, that, what's her name, Moira Rose, when she performed the wedding at the end of the show, a 2-part hat, yes, go stream it if you want to. A 2-parted hat like this is called a mitre. That's where the name comes from. It's like that hat that a bishop would wear, a mitre, so it's the bicuspid or mitral valve. So mitral valve is the most common name you're going to see in the medical world. tricuspid for this one. The other valves are the two semilunar valves, aortic and pulmonary. The aortic semilunar valve, you cannot actually see on this model. See why I have to use a bunch of models. I would have to go way up here with my fingers until I would finally find it, and I don't really see it there unless I open this up and go way in here, and they don't even really have one. So see that? It does have, however, this model, Jumbo, a perfectly good pulmonary semilunar valve I can see right here. This pulmonary semilunar valve guards the entrance into the pulmonary trunk. So 4 valves, tricuspid, mitral, pulmonary semilunar valve right here, and on this model I can't even see the aortic semilunar valve, but trust me, you can see these valves better on some of the other hearts. Like this one right here. So let's look at the great vessels on this model. Notice coming across the front at a very steep angle, I have the pulmonary trunk. That's the most anterior of the great vessels. The pulmonary trunk, which splits into pulmonary arteries right here on the backside. The aorta coming out, making its arch. Now, I know what you're thinking. Dwayne, how would you ask this on a quiz or a practical? If I wanted the name that you produced to be the aorta, I would have to put a dot, say right here, and say name this blood vessel, right? Aorta. If I wanted the aortic arch, I would have to put several dots, like so, across the arch and say, name this structure. Aortic arch. See the difference? Pulmonary trunk, pulmonary arteries, aorta, aortic arch. Here I can see the pulmonary veins in red coming back into that left atrium. And if I keep going around, very nice here, the superior and inferior vena cavas. Superior, inferior vena cava. Now let's take a look at these valves in this open face heart, which here I can see them a little more clearly. So see the right atrium here, left atrium there, and the valves that separate the atrium from the ventricle below, the AV valves, right here is the tricuspid valve with 3 cusps or flaps. Right here is the bicuspid valve, which has 2, or mitral valve. So again, with the valves, mitral valve, tricuspid valve. This is what everybody's going to call them. Mitral valve, tricuspid valve. I also have, at the bottom of this pulmonary trunk, a very nice pulmonary semilunar valve. And at the opening of the aorta, see how I pivot? This is the left ventricle, the aortic semilunar valve right there. Aortic semilunar valve, pulmonary semilunar valve. The semilunar valves get their names because they're half moons. The cups here are like a half moon. So think of a crescent moon sort of cup there. They don't have these cords and big papillary muscles below like the 2 AV valves do, the tricuspid and mitral. So pulmonary semilunar valve, aortic semilunar valve. Let's look at another model, because remember I have a bunch of models. Let's look at the great vessels first. As always, kittywampus across the front, pulmonary trunk, which branches into its pulmonary arteries going to each lung. Pulmonary artery, see that? Keep spinning it around. There's the other pulmonary artery right there, shooting to the lung. The aorta and its arch, right here. And pulmonary veins right here in red. Remember, carrying oxygenated blood back from the lungs to the left atrium. Pulmonary trunk, pulmonary artery, aorta, aortic arch, superior vena cava, inferior vena cava. Notice, even though the models are different, the basic layout is the same for each one of these. If I open this one up, I can see the sort of rust-colored mitral valve separating the left atrium and ventricle, tricuspid valve separating the right atrium and ventricle, pulmonary semilunar valve, and aortic semilunar valve way up in there. See that aortic semilunar valve way up in there. Great vessels, 4 valves. Let's look at this one. Again, I'm just taking you through the same thing, so I know it's repetitive. That's good. Repetition is good here. Pulmonary trunk, pulmonary arteries. Now, in our first online labs that we had roughly a year ago, I had to make those a little faster, and one criticism I got from students was not enough views, not enough angles. So I'm taking you through all these models. It takes more time. That's all right. You can pause me. You can fast forward me. You can take your time here and go as fast as you like to, but a lot of repetition is a good thing. Pulmonary trunk, pulmonary artery, aorta, and there's the aortic arch. So this whole thing is the aorta, which actually does a U-turn and runs right down in your thorax into your abdomen. So the name of the blood vessel, 1 dot, aorta, multiple dots on the arch, structure, aortic arch. I can also see my superior and inferior vena cavas here. Remember, these connect to that right atrium. If I stick my marker up there, see it come out, so now I'm in the right atrium. This is one of the vena cavas. Superior, inferior vena cava. And last but not least on this model, the pulmonary veins in red. See that? 2 pairs coming back from the lungs, dumping this oxygenated blood into the left atrium. Now let's talk valves. Very nice on this particular model. I can see the 2 very serious looking mitral and tricuspid valves right here. These are the ones with all these little strings or cords on them. Mitral, tricuspid valve. Again, mitral valve separating the left atrium and left ventricle. Tricuspid valve separating the right atrium and right ventricle. And then my 2 semilunar valves, yes, you can tell. Pulmonary semilunar valve and a very nice giant aortic semilunar valve right there. Pulmonary at the base of the pulmonary trunk, duh, and aortic at the base of the aorta. Great vessels and heart valves. One more model. The last one for these structures, which is just like the one you saw earlier, just a little larger. So let's go through our great vessels. Pulmonary trunk, separating into pulmonary arteries. See those? Pulmonary artery, pulmonary artery. Aorta, aortic arch. Superior and inferior vena cavas. Superior and inferior vena cavas. And pulmonary veins down here in red, bringing that blood back to the left atrium. And the valves, we have, with the strings, the cords visibly apparent right here, the tricuspid valve right here, the mitral valve, pulmonary semilunar valve at the base of the pulmonary trunk, again, duh. And I have to angle to see it. There it is, the aortic semilunar valve, right there. Aortic semilunar valve, pulmonary semilunar valve, tricuspid valve, mitral valve. Now, would it be preferable for you to be right here getting your hands on these models? Certainly. But guess what? You're not here. So I show you all 5 models to give you all the views possible. So watch this over and over again. From the different angles, different sides, all different models. Okay, next up in our lab manual, still on page 6, because look, there's a whole bunch of cardiac structures here. Now we're to the section on the coronary arteries, coronary veins. Not the great vessels, but the arteries and veins, red for artery, blue for veins, that actually lie on the surface of the heart that supply blood to and carry blood from the myocardium. Remember, these are cardiac muscle cells, so they have big energy demands, lots of glucose needed, lots of oxygen needed. These cells are very good at doing aerobic metabolism, rather crappy at doing anaerobic metabolism, but that's lecture. I don't want to get too much into lecture for you, because you get big fun in your online lectures as well. But what I will do is show you these coronary vessels as they lie on the surface of the heart. Remember, and hopefully you already made a picture like we described, because in these grooves that we find on the heart, we see some of these vessels living there paired together. You can see what I did in my lab manual was I linked those two that were paired. If I would have had enough markers with me right here in lab, I would have used 3 different colors. The ones that live together in the different sulcuses of the heart. On these 2 models in particular, I can see a very nice anterior interventricular sulcus. This groove right here on the anterior surface of the heart. And what 2 great vessels do I have living there? Red, the anterior interventricular artery. That's the whole red one. Yes, I know it branches, but for you it's just all the anterior interventricular artery is the red one. The blue one, the great cardiac vein. Those are the 2 in this sulcus. Anterior interventricular artery, great cardiac vein. Anterior interventricular artery in red. Great cardiac vein in blue. Yes, it branches apart. This is what they do. Actually, the veins go the other way. Anterior interventricular artery, great cardiac vein. Oh, I know you have it already, but a little repetition never hurt anybody. And guess what? You can mute me. How often can you mute one of your professors? You can mute. Anterior interventricular artery, the red one. Great cardiac vein, the blue one. Let's not forget jumbo. Look at these monstrous things. Anterior interventricular artery in red, great cardiac vein in blue. And this one serves to illustrate something that might help you out a little bit in lecture. So the epicardium, or sometimes called the visceral pericardium, is a covering on the outer surface of this heart, and a little bit of adipose, sometimes some loose irregular connective tissue, essentially areolar connective tissue for you 223 throwbacks, serve to hold these vessels on the surface of the heart. So they're not just flapping around here, they're actually held in place by connective tissue, usually a little bit of adipose and loose irregular connective tissue just under that epicardium, which would be a squamous layer right here. Anterior interventricular artery, great cardiac vein. Now let's look at the reverse side, the posterior side of these heart models. In the posterior interventricular sulcus, which two vessels do I have living there? I have the posterior interventricular artery, right here in red, and the middle cardiac vein in blue. Posterior interventricular artery, middle cardiac vein. These are the pair on the back side, the posterior side of the heart. Because you have no idea which model I will choose. That's the beauty part for me on a practical. We need to see this on all of them. So this model right here, notice where we are. This is the back side of the heart. There's the anterior surface, posterior surface. What do we have right here? Posterior interventricular artery, middle cardiac vein. Kind of a weak posterior interventricular artery on this model, but that middle cardiac vein's certainly nice, isn't it? Look at that thing. This one, let's flip it around. Right here, yes, you can see posterior interventricular artery in red, middle cardiac vein in blue. This one, anterior side, posterior side. Look at these beauties. Posterior interventricular artery in red, middle cardiac vein in blue. Very, very nice on this one. How'd you like that? Just make sure you're awake with chalkboard noises on the lab tables. They always make that sound. All right, this one's the same as that smaller one. On the back side here, I see the posterior interventricular artery here and middle cardiac vein right there. And you say, Dwayne, what about these two over here? Do they have names? Well, sure they do, but you don't have to care what they are because nobody's going to quiz you on those two. On the back side, you have the posterior interventricular artery and the middle cardiac vein. The other pair are the ones on this right lateral side. The ones on the right lateral side. They are the right marginal artery and the small cardiac vein. Let me point them out to you on each model. Right marginal artery right here where my pointer is, and small cardiac vein right there. These two are on the, remember he's facing us, the right side along the margin of the heart. Small cardiac vein in blue, right marginal artery in red. On the jumbo model, so I look over here to the right side, turn it, right marginal artery, sometimes called the right marginal branch, and small cardiac vein. This model, look to the patient's right, margin of the heart. What do I see? Right marginal artery, small cardiac vein, partners. Right marginal artery, small cardiac vein. Here, turn. Right marginal artery and small cardiac vein. This model, turn it. This one is probably the weakest for these two vessels. I can see the small cardiac vein, and this is the right marginal artery. Not real great on him. I'm just telling you straight up, I would not use this particular model for the right marginal artery because his right marginal artery is not very good. Now that's not all the cardiac vessels, is it? We have a few others. If I look at my handout, the right and left coronary arteries, circumflex artery, and coronary sinus. These are coronary vessels I find in primarily the coronary sulcus. Remember that area where if I was strangling this heart, my finger would go right there between the atria and ventricles. That's the coronary sulcus. So let's take a look at these remaining coronary vessels. First, I'm going to show you the limitation we have for the left coronary artery. Left coronary artery. This is the left side of the heart. This is the right side of the heart. The left coronary artery usually is found nestled right under this flappy structure called an auricle, and on this model I can't really distinguish it at all. I'm going to say that again. On this model I can't really see the left coronary artery at all, at least not worth anything. On this model I can see it a little bit. On this model I can see a little bit of it because do you see right here where this artery branches, or in vascular speak I would say bifurcates, it splits. Before it splits right here, this is the left coronary artery, which splits into the anterior interventricular artery, and this one sweeping around within the coronary sulcus called the circumflex artery. but only a small bit, I'd say probably less than a centimeter on this particular model. This really old-fashioned model, which some people wonder why we even have it here, this is one of the reasons we have it, has a fairly decent left coronary artery, see it right here, which branches into the anterior interventricular and circumflex going around. So left coronary artery, not bad on this one. Not too bad on jumbo either. This is the circumflex artery, right here in red. This is the anterior interventricular artery. If I remove that top section of the model, I can see a very good left coronary artery. So as you're trying to position this thing in your mind, and this left coronary artery shoots right out of your aorta, this left coronary artery branches to become the anterior interventricular artery and the circumflex artery. Left coronary is before it branches. So this one has a decent section of left coronary artery right there. And this last model here, which is just a bigger replica of this one, doesn't really have any left coronary artery to speak of. Most of these models have a fairly decent, let me turn it, right coronary artery. So I want you to imagine this aorta shooting out of your left ventricle, and as soon as it hits the surface, I've got two big arteries that come out of it, a left coronary artery and a right coronary artery. So the right coronary artery has a much longer exposed run on a heart. It's this red one, which then has a chute going down called the right marginal branch or the right marginal artery. So this is the right coronary artery under my finger, the red one. Orient your heart. See the pulmonary trunk? Right coronary artery. Pulmonary trunk. Right coronary artery in this coronary sulcus. Great big right coronary artery right down here. Right coronary artery, the red one. And on the giant model, I'm just putting him back together. That's the sounds you hear. Notice here a right coronary artery as well. Right coronary artery. Now, I already mentioned, but I'll show you quickly, the circumflex artery in the coronary sulcus on the other side. Remember, the left coronary artery isn't that long, and it branches. So this is the circumflex artery, left side of the coronary sulcus. Circumflex artery, the red one. Circumflex artery. Not a real good one on this model. Notice that it's sort of buried. You can't see much of it. Just making sure you're awake. See the circumflex artery right there in red? Circumflex artery. Pause, rewind, draw these out. Many, many times that's the thing to do. Last but not least, we have a coronary vessel that doesn't have artery or vein in its name the way most people say it. And that's this big blue area right here. See this bulge my finger is on right there? On the posterior side of the heart. This is called the coronary sinus. A sinus is a chamber, a collecting area. So this blood in the veins comes back, gathers up here before it goes into the heart in this little chamber or pocket called the coronary sinus. Let me show it to you on the other models. On jumbo, coronary sinus, right here, this bulgy part on the back side of the heart. So see that middle cardiac vein coming up? It dumps right into the coronary sinus. This heart model, coronary sinus, quite large here. The coronary sinus on the backside, posterior side of the heart. And I think that's all of them, because remember the other was the same. This one probably the weakest coronary sinus right here. It's actually a larger bulge of a vein right there, so coronary sinus right there. Okay, so here we once again have my quintet of heart models. Remember, two of them, number 2 and number 5 here, as you look at them panning across, are really just different sizes of the same model. But we still have a few cardiac structures from the models that we have to go over this last section on page 6. Now, I've already hit 3 of them. You can see I put little dots next to mine to keep track. The anterior interventricular sulcus, coronary sulcus, and on the backside, posterior interventricular sulcus. We already know those. But this is sort of hitting some of the parts of cardiac anatomy that we haven't hit yet. So, keep track and follow along with me. Let's look at the apex of the heart. Now, as I look at these 5 hearts, you can see that the heart has almost an arrowhead shape. It definitely has a point on the inferior side. The base of the heart is up here where the great vessels go in and out. The apex is the point of the heart. So for each heart, I have 1, 2, 3, 4, and 5, an apex. So the apex of each heart is the point. The heart also has a structure called an auricle on it. In fact, they have essentially 2 auricles, though sometimes people only use it for one of the 2. The auricle is this flap-like structure that I see hanging off of an atrium. I'll say that again. The auricle is this flap-like structure I see right here. See that right there? Hanging off of an atrium. I'll show you slightly different views. So here I have an auricle, see the little flap right here. On the right atrium, here I have an auricle on the left atrium. I am actually old enough that I can remember when people used to refer to the 2 atria as auricles. Don't do that anymore. So I'm going to shake my finger at you. Don't do that anymore, because now in the 21st century, we say atrium. So the auricles are these flaps. Just like you had an auricle on your ear, we have auricles on our heart. Now we have some structures associated with my valves. Say that again, some structures associated with my valves. Let's look at this one first. This small heart model. And I will zoom in on it a little bit for us. And take off the anterior half. So the two valves that I see here, the atrioventricular valves, the tricuspid and mitral valve respectively, these valves have tendinous cords that I can see here, these strings attached to the flaps of the valves. These things are called the chordae tendinii. You pronounce it as if there are two long E's at the end of the word. Notice the spelling. Chordae tendinii. Many people in the U.S. try to make two words that rhyme, and they say the chordae tendinae, or they just cop out altogether and say chords. Doesn't really matter how you say them. It could matter in your future how you spell them, and definitely if you recognize the correct spelling on your lab quiz. So these strings are the chordae tendinii, and they are attached to these projections off the surface of the endocardium. See this little bump here, this little bump there, here's one over here. These things are called papillary muscles. Papillary muscles. The tendinous cords come down and attach to these papillary muscles to actually pull down on those flaps to keep those flaps from flying open in a backwards direction. I'll mention that again in lab, and you'll hear about that in your lectures, I'm sure. But we need these papillary muscles to pull on those cords so that the valves don't fly open the wrong way. They actually would. Let's see these same two structures on the other models. So this one, if I open it up, I can easily see the cords here, chordae tendinii, attached to the papillary muscles, a little bit weak on this model. The cords are fine, the valve is fine, the mitral valve there, but the papillary muscles are a little weak. Let's look at this one. Take off its face. And here they made a much more delicate model. They actually represent the cords as little strings. See these strings, almost like parachute strings. So the chordae tendinii, beautiful papillary muscle right there. Papillary muscle right there. Papillary muscle, chordae tendinii. Now let's look at jumbo. Right here. So I can see the cords, chordae tendinii, right here, and papillary muscle at the base of the cords, papillary muscle. Chordae tendinii and papillary muscle, actually considered modifications of the endocardium. Next up in our list, we're jumping around a little bit, but that's okay because we're here. I want you to look at this wall of myocardium right here that separates the two ventricles. So jump down I don't know, maybe 2/3 of the way down that list on the far bottom right side of page 6. Here we have the interventricular septum. Septum is a wall between the two ventricles, interventricular septum, separating the left and right ventricle from one another. Interventricular septum. In this model, right here between my fingers, the interventricular septum, wall that separates the two ventricles. Let's get another one, because I know you love all the different models that I can use. I say that sarcastically. Right here, interventricular septum. Interventricular septum. That one's pretty easy. I don't think you're going to miss that on whatever model I show you. We also have an interatrial septum, that separates the atria from one another. Not particularly well represented on most of these models unless you open the thing up like so. This wall right here is the interatrial septum. Ooh, squeaky squeak. Right here between my fingers is the interatrial septum. So most of these models you sort of have to open it up to see that. This model right here, interatrial septum between the two atria, between my fingers right now, interatrial septum. And I happen to notice a couple other structures in this model right here that are very well represented. In your list, I want you to find two names: the fossa ovalis and the opening of the coronary sinus. I'm going to say that again: the fossa ovalis and the opening of the coronary sinus. You might remember from way back in 223 that a fossa is a depression or a dent in an anatomical structure. We have fossas in some of our bones, you might recall. So I want you to look at this dent where I'm putting my thumb. Right there, it's about a thumb-sized dent in the wall of the right atrium, everyone. This structure is called the fossa ovalis, the oval fossa. During fetal circulation, this was actually a hole between the two atria called the foramen ovale. You may learn about in your lecture. In an adult heart, it's just a depression, the fossa ovalis. Now, this will freak some of you out, but that's okay. That's part of my job. In some of us adults, there's still a hole there. It's just the force of the blood holds the two flaps of the interatrial septum together like that, so you never know that you have a hole there. In most of us, it sort of seals up and becomes this fossa ovalis. Fossa ovalis. And this hole, where I'm putting my probe right now, this hole in the right atrium again, is the opening of the coronary sinus. This is where that coronary sinus, you may recall, it's under my thumb right here. This is where that coronary sinus dumps blood into the right atrium. And what they do on most really nice models is the model builder just throws a little splash of red paint right there. So inside the right atrium, I see a fossa ovalis and the opening of the coronary sinus. I think I can see this same pair of structures on other heart models. Fossa ovalis, right there. Opening of the coronary sinus with a little splash of red paint. Again, inside the right atrium. That's where you see these structures the best. Let's look at this one. Open up that right atrium. Oh, look at that. Beautiful. Fossa ovalis and opening of the coronary sinus. Some models don't even bother to show you the opening of the coronary sinus, but these three that I just showed you actually all have very good fossa ovalis representation and opening of the coronary sinus. Now, if you look at the list, you will notice that that's not every single thing. We also have one last, I believe, final structure called the trabeculae carnii. The trabeculae carnii, and yes, two E's on that one too. Trabeculae carnii. What are trabeculae? I want you to think back to 223. Trabeculae are an irregular network, shards, pieces of something. Think of the trabeculae in your spongy bone as an example. This is a name we often reserve to describe the irregular ridges, see this, in a ventricle. You can hear my probe bouncing across that roughness. Not the papillary muscle. These ridges in here are called trabeculae carnii. I can see them very nicely on this model. He doesn't have good papillary muscles, but look at those trabeculae. Right there, right there. Trabeculae carnii. Your lecture professor can tell you what people theorize they are for. Nobody's quite sure, but we do have them. You can see it right here. Look at these ridges. Trabeculae carnii. Now, for those of you with a Latin-based language at your mental fingertips, if I go to a restaurant, only getting takeout, of course, and I order carne anything, what am I ordering? Meat. Trabeculae carnii. These are trabeculae made of meat, not of bone or some other structure. So look inside the ventricles of this heart model. See all these ridges here. Hear my probe going across them. Not the papillary muscles, not attached to cords, but these are trabeculae carnii. Trabeculae carnii. If I look at the inside, the faceplate that I have removed from Jumbo here, look at all those trabeculae that you see there. Trabeculae carnii, the meaty trabeculae, everyone. And I think that is every structure on the list, and wasn't it fun going through them? There is one last remaining structure, and I want to make sure I point it out to you on my three models that show it very well, because I don't want any of you people jumping all over me, all panicked. This is panic hands, all panicked about me not showing you this one. This is the ligamentum arteriosum. The ligamentum arteriosum. This is a ligament that connects two arteries, namely pulmonary artery and aorta. It's this white thing right here, which right now in this model is just a bunch of collagen fibers holding these two arteries together, the ligament arteriosum. back in our fetal days when we were all happy living in utero, this was another fetal holdover like the fossa ovalis we saw before. This one, once upon a time, was called the ductus arteriosus and was a shunting of blood from pulmonary circulation to systemic. I say that for lecture reasons. Now, after we're born, it seals up pretty quickly. So this structure right here connecting the aorta and the pulmonary artery is the ligamentum arteriosum. I can see it right here on this model as well, white, ligamentum arteriosum. And on our friend Jumbo, right here, ligamentum arteriosum, connecting pulmonary artery and aorta. Ligamentum arteriosum. Our next little unit of study is the sheep heart dissection. So notice this small section of your lab, page 7, talking about the dissection of the sheep heart. Now I will space it out and time it in this online lab so that you see the models before you see the actual sheep heart. I always recommend that very strongly to students when we are live in person. Always master the models first before you look at an actual dissection. That makes your life a lot easier, hopefully makes your learning of all the structures a lot faster. Now keep in mind that only these structures right here on page 7 am I allowed to ask you from a sheep heart dissection. So many of the structures that you are responsible for on lab models, because they're designed, handcrafted, painted, are much easier to distinguish than on an actual sheep heart. So I'm limited only to this small list. Remember that as you watch me do the sheep heart dissection. Since for these online labs, I am not only the writer, producer, director, I am also the cameraman, so I will do my best to create video images for you as I do the dissection. But if I get in the way or it's a little fuzzy, something like that, make sure that you stay calm, because in the practice section of this online lab, I will endeavor to create good heart images for you to use to practice for your lab quiz and practical after that. So, without further ado, why don't you, at least in the figurative sense, glove up with me and let's cut up some dead stuff. So what we're looking at here are two sheep hearts, one rather larger, one rather smaller sheep heart, and I threw in a heart model right there in the same exact orientation. Now remember, for your study, it's just this list right here of structures that I'm allowed to ask you in this sheep heart dissection. And consulting the list, you will notice that there aren't really many external structures. You don't see any apex, you don't see any coronary vessels. It's only these structures that are fair game for that lab quiz, which aren't really structures that are that difficult for us to see or find, with the possible exception being the great vessels. Those vessels that are actually connected to one of the chambers of the heart. So I'll do a little camera work, a little zooming here to try to show you some of these structures. So let's look first at this slightly smaller heart that you see. Maybe you can notice that this is the apex, not that it's on the list for the sheep heart, but here's the apex. I can see great vessels up here. They're always cut fairly short in these prepared sheep hearts. There are many ways that you might see people dissect a sheep heart, but this is my way. And let me take a quick poll of all the other A&P professors that are in the room with me. Does anybody have a different way that they would like to dissect this heart? Oh no, so I guess Dwayne has it. So here we have my preferred method of sheep heart dissection is just a cut down the front of the heart, actually up the heart, so it opens in this fashion, sort of like the hood of a car. And I can see quite obviously right off the bat the two ventricles. I always look at the heart this way to orient myself. I can tell this is the left ventricle. How can I tell so quickly? Well, look at the thickness of this myocardium right here as compared to the thickness of this myocardium over here. Always use the thicker myocardium in the left ventricular wall to orient the four chambers of the heart. Once you know this one is the left ventricle, then identifying the other chambers gets pretty easy, doesn't it? So if we start looking at a few things that we can see right here, let's look at the layers of the heart wall. So right here at the tip of my probe, you can see that it's slightly darker here. This is where the epicardium is, holding those coronary blood vessels down, the epicardium or visceral pericardium. If I close the heart back up, I can see the impression of where some of these coronary vessels are, but they haven't been stained for me. They haven't been painted by some model builder. So you're not responsible for identifying them. You can see connective tissue, a little bit of adipose right here. This is just in and under that epicardial layer. So just like on the models, we have the epicardium right here, myocardium, which is the cardiac muscle tissue, and then I can see a little bit shiny right here, smooth looking, nice, slick, simple squamous epithelium. That's the endocardium. I can do the same thing over here in the right ventricle. Epicardium, the outside, myocardium, much thinner here, and endocardium, nice and shiny on the inside right there. So always using one of the ventricles for the layers is pretty standard because it's just easy to identify them. So epicardium, myocardium, endocardium. I could do the same exact thing with this larger sheep heart. See, I've already dissected this one, so it opens like the hood of a car. And I see the much thicker left ventricular wall, much thinner right ventricular wall to orient me. This right ventricle is pumping blood to the lungs and back. And in case you didn't think about it, remember your lungs are located just on either side of your heart. It's not that far away as compared to the left ventricle, which has to pump blood into the aorta, into systemic circulation you'll talk about in your lecture, which means everywhere. That's not the lungs. So a much greater distance, a lot more myocardium right here. So epicardium, I can see just because this has a little bit of adipose, it's a little lighter right there. See that? Epicardium, myocardium, and then the inside surface that would actually be touching the blood, endocardium. Epicardium, myocardium, endocardium, the three layers of the cardiac wall. Other structures that are pretty noticeable here would be the four chambers which I think I can identify pretty easily. I've already done 2 of them, haven't I? This is the left ventricle. So I indicate this on a quiz, and I say name the chamber. This one's the left ventricle. Again, the thick myocardium here. What's this chamber? Right here, the right ventricle. Certainly, easy to do. Then I want you to notice these cords. You might remember the name, and we'll refer to them again in a few moments. The chordae tendinii, attached to papillary muscles. Now, I bring this up for a particular reason. In your list, you see chordae tendinii are included. Are the papillary muscles anywhere in this list? No. So I cannot ask you papillary muscles, can I, on the she part. I'm just pointing it out to you because I can see them stuck right here at the bottom of the cords. The reason I show you the chordae tendinii right now is because they're attached to the flaps, remember, of the valves that separate the atrium from the ventricle. The AV valves, or in this case, on the left side, this is why we do the models first. I'm shaking my probe at you. This is why we do the models first. This one is the what? Mitral valve. So I follow the cords up right to the mitral valve here at the top of the left V-shaped ventricle. What would I see at the top of the right ventricle as I expand it with my hands here? Can you see the cords in there? See the cords? And the flap, this would be the tricuspid valve that my probe is on right here. This is the tricuspid valve separating the right atrium from the right ventricle. So I can see those 2 valves pretty easily on this heart. And this larger sheep heart, I can see the same. See the cords? Follow them in a superior direction, and this is a flap, one of the flaps of the mitral valve. Right there. This valve is the mitral valve. Look at the cords. Over here, if I expand this out, I can see some cords here. They're a little bit chopped up, going up to the tricuspid valve, which is right here, separating the right atrium from ventricle. Look at the back side up here. I can see those cords, see how it closes. These are the cords and the other flap of the mitral valve. So the 2 valves that separate the atrium from the ventricle on each side, remember, I'm going to see those cords attached to them. So the chordae tendinii right here, separating or anchoring rather the mitral valve's flaps. And then watch my probe, everyone. If I simply move my probe up through this valve, the chamber that I am in right now, see me moving it there probably, right there, this is the left atrium, right here where my probe is wagging around, above that mitral valve. Everybody look, this is the left atrium. I'll try to expand it with my fingers. There it is. See that endocardium right there? The left atrium. Right there. I'm doing the best camera work I can here. It'll be a little more obvious, I'm sure, in the practice section, but I want to do my best here. So as I expand this out, left atrium, through the valve, left ventricle. Over here, the right atrium is always a little tough to get to, not with my probe, that's pretty easy. So everybody look, see the tip of my probe moving around this auricle on the right side. So the right atrium is up in here. There's a big empty space that my probe is waggling around in here. Let's look at the other heart. This is why we always try to use more than one, so a person can see the structures a little differently. And as I wag my finger at you, remember, when you study this, don't just only use my images here. Use your atlas, use MH Connect, use online image searches, find some sheep hearts, dissected one way or another. The more images, the more different images you see, the easier, the faster this will be for you. So as I open up this one, just because of where it's cut, this one's a little easier. So left ventricle, right ventricle, cords, mitral valve, and then above it, right here, this is literally it, where my probe is. This is, let me move this little flap of myocardium out of the way. This is the left atrium, right here. Ventricle, atrium. The right atrium, always a little tougher because of dissection. So I see those cords, and I stick my probe up above this chamber that I'm waggling around in right here, above the tricuspid valve, is the right atrium. I can also notice here, between the two ventricles, this interventricular septum. Right here. This one's shredded a little bit here and here. That's just a remnant of my dissection. Here I can see it a little cleaner in this smaller heart. Interventricular septum. Right here. The interventricular septum. And what would I call the septum, the wall, that separates the two atria from one another in this heart? So above, I'm stabbing it right now with my probe. It's between my probe and my finger. This is, in fact, let's do this. I'll even get a second probe. Right here, so notice I'm above the corded valves. Right here is the interatrial septum. The interatrial septum separating the left and right atria from one another. On this larger heart, again, just because of where it was dissected, I can actually see the wall right here, separating the two atria from one another, the interatrial septum. The interatrial septum. So we've got the layers, we've got the chambers, 2 of the 4 valves, and then the great vessels. So let's look at the other 2 valves that are in this heart. You might recall those would be the semilunar valves at the base of the great vessels. Now, I mentioned this before. You can see a couple great vessels, usually pretty easy to see, at the top of this heart, but they cut them off very short. Notice I don't see any aortic arch. I don't see any pulmonary arteries shooting off the pulmonary trunk. And it is so common, this appearance, look at this one, how short it's cut off. No arch, no branching of the pulmonary trunk. it can be very, very difficult to find some of these great vessels and the valves associated with them, but you might guess it, I do have a technique for this that shows me the great vessels and the valves associated, the semilunar valves. I'm going to use a pencil for this. Why? Because I found a pencil here in the lab. Sometimes I use a pipe cleaner. Sometimes I use a metal probe. But today, I have a pencil, so I'm going to use a pencil. And then, of course, this pencil is going to go in the garbage after this, except for whatever photos and images I might take with it for your practice section. So notice these two great vessels at the top side of this particular heart. What I'm going to do is stick my pencil into one of them and see where it comes out. So I find a great vessel, I cram my pencil into it, and notice my pencil just goes somewhere, doesn't it? Where is the tip of my pencil, the eraser end? What chamber of the heart is it in? It's in the left ventricle. So this pencil has gone through a great vessel, through a semilunar valve, and into the left ventricle of the heart. What great vessel exits the left ventricle? I'll wait. You can pause me. That's the aorta. So the great vessel I am in right here is the aorta. The only way you would know that, looking at this sheep heart, is due to the fact that the pencil tip ends in the left ventricle. So if you saw an image like this, with a pipe cleaner, probe, this very pencil, in the left ventricle, exiting through a great vessel, and the question was, name the blood vessel, that's the aorta, isn't it? So the aorta is connected to the left ventricle. Let's try it on the larger heart, shall we? Same idea. So I look at these great vessels on the back. I have a pretty good idea that this one right here is the aorta, but I'm not positive until I cram a pencil down there. Now, watch what happens as I stick the pencil into this thing. Notice how it is forced to skew left, which it just did. Going down into what, everybody? The left ventricle, right into the left ventricle. So this pencil is in which great vessel? The aorta. This is how you will know. You will look at the dissection. You will look at what chamber this thing ends up in. And let me zoom out a little for you. This is what tells you the great vessel that you have is what chamber the pencil or the pipe cleaner or the probe ends in. This is the left ventricle right here where I'm tapping with my thumb. So that makes this great vessel the aorta. Let's try it with the other one on the front side of the heart. This is the great vessel I just stuck my pencil into. Here's another one. Notice it's the most anterior. So I have a good suspicion what this one is, but I'm going to go ahead and wiggle my pencil into it. And notice this one suddenly skews to the right. Remember my wording from before. This great vessel goes kitty wampus over here to the right, and I can feel, you're not here so you can't feel, I can feel that I've just entered a chamber. So as I open this up, where's the tip of my pencil? Look at the thin wall, in the right ventricle. In the right ventricle. What great vessel exits the right ventricle? The pulmonary trunk. That makes this the pulmonary trunk. So the only way you really know that is because of where the pencil ends up. Let's try this one. So I've got these great vessels here. I see this one. I'm suspicious because it's on the front surface. Let's see if I can get my pencil in there in this smaller one. Oh yes, look at that, kitty wampus on the front or the anterior surface. And I keep wiggling and oh look, I just got into a chamber. Where's that pencil tip, everybody? In the right ventricle, which makes this the pulmonary artery. I hope everybody sees that. Pulmonary artery, or since it hasn't bifurcated or branched yet, we say pulmonary trunk, which is one of the great vessels. So pulmonary trunk, the pencil is in the right ventricle. Aorta, pencil is in the left ventricle. And the pencil has passed through the pulmonary semilunar valve, which is the hardest of these semilunar valves to find, but it's right up there. The aortic semilunar valve isn't very difficult. Everybody watch. If I go back through the aorta, which forces me left again, right here, see the tip of my pencil? Right here, this is the aortic semilunar valve. Right there's another flap for it. Notice, no cords on this little flap. This big thing right here is the aorta. Now notice it's right next door to the mitral valve. So look over here, see this that I'm pushing with my pencil? No cords here. Cords over here on these flaps. So they're continuous pieces of connective tissue, but no cords over here. Aortic semilunar valve. Let's look at the other heart, a little bigger. If I stick my pencil again through the aorta, notice it's skewing to his left. This thing right here, look at where the eraser is, this is the aorta. And at the bottom of it, right here, I'm moving one of those semilunar flaps. This thing, hopefully you can see it. And again, if the camera work isn't that great, make sure that you're paying careful attention in the practice section. No cords. This is the aortic semilunar valve. Left ventricle, follow it up. Aortic semilunar valve. In this same heart, if I go back into the pulmonary trunk, right here, everybody, notice I'm directed to the right. This is just the way the pencil actually goes. His right, not your right. So I come out in that right ventricle. The pulmonary semilunar valve is back here at the top of the right ventricle. Usually not well seen in a dissection. In fact, you don't see that many people asking pulmonary semilunar valve because it's so difficult to see in this dissection. But the other three are pretty obvious. Mitral valve, look at the cords in here, everybody. Tricuspid valve, a very nice shot right here at the tip of my pencil. of the aortic semilunar valve. The other great vessels we have are the superior and inferior vena cava and the pulmonary veins. These are the great vessels attached to the two atria of the heart. Pulmonary veins going into the left atrium, vena cavas going into the right atrium. But again, we have a problem with, first, the nature of a sheep, and second, the way these things are cut. So I want you to look at the top of this heart. I can see holes up here, but it's very difficult to tell where some of these holes are going. See, there's a great vessel is cut right here. See this one? But I have no idea where that goes until I cram a probe in there. And now I notice, everybody, look at the pencil, look at the angle. I'm going to move it around in here a lot so you can see. Am I in the right or left atrium? This is the right atrium that I'm in, obviously. So that makes this one of the two vena cavas. But on the sheep heart dissection, these things are always cut so short, there's really just one vena cava. There's not really a superior and an inferior, and believe it or not, the sheep doesn't have one because the sheep is out in the pasture. Head would be over here, tail's over there. It actually has an anterior and a posterior vena cava that come in, and then they cut it off as soon as they sort of merge into just one great big vena cava. So we don't have an anterior and posterior vena cava on most sheep hearts. We just have the two of them together. So if I'm connected to the right atrium, as I am here, again, look at the angle of that pencil, that's the vena cavas. Let's check this one out just at random. Looking for a hole up here, not the aorta. So I see several holes up here, but I'm not sure which one to go in. I guess I'll go in this one that's farthest back, and oh voila, look, I'm in the right atrium here again, so this is the vena cava. But I want you to look closely, people. Look how short it's cut. There is no way to tell one from the other, so on a lab quiz, what do I have to do? If I have it here going into the right atrium, see the tip of the pencil or the eraser? If I'm in the right atrium, what answer would you select? Superior / inferior vena cava. I'd have to include both of them in the name. I couldn't have superior as choice A, inferior as choice B. There's no way to tell on this sheep heart dissection because they cut it off too short. So this is the way it would be. Remember this for your quiz. If I'm in a great vessel that goes to the right atrium, the answer is superior / inferior vena cava. You can't really distinguish on these two hearts that I have. The last great vessel, the pulmonary veins, I would expect to be something that goes directly into where? The left atrium right here. What great vessel is my pencil through right now? Pulmonary veins. This is the left atrium, remember. Right there, left atrium. Pulmonary veins going into the left atrium. Let's see if I can find something similar in this heart. Oh, look, there it is. Right here. Look at my pencil, see where it is. If I open this heart up to see the tip, See the tip of the pencil poking through right there? What great vessel would I be in here? The pulmonary veins. Hopefully the practice section will reinforce some of this and make it a little more obvious to you, but remember, for the great vessels, what chamber of the heart, which of the 4 chambers is the probe or the pencil in, tells me. Left ventricle, the great vessel is the aorta. Right ventricle, great vessel is the pulmonary trunk. Right atrium, great vessel is the 2 vena cavas. Left atrium, the great vessel is the pulmonary veins. Again, I apologize if there was bad camera work here. Trying to do the best I can, but I can make a concerted effort, how's that, for the practice section of this online lab. And as you look at these 2 hearts here, let me just give you a free piece of advice. When it's Valentine's Day and you're feeling all romantic and you want to send somebody a heart, do not send them one of these. That's just creepy. In fact, don't even send them one of these. That's only slightly less creepy. Just get them a box of chocolate or something, will you? Okay, everybody, next up in your lab manual, you have a relatively brief section on heart sounds. Those things people typically listen to through a stethoscope. As you can see in your lab manual, these sounds are called Korsakoff sounds. You typically do not pronounce the T there, so Korsakoff sounds. And a couple of things people often mess up a little bit is, 1, what causes these heart sounds, and 2, how do you actually listen to the heart sounds? So first, let's talk about what causes these sounds that you hear through your trusty stethoscope. The sounds created are not, I repeat, not N-O-T, created by the heart beating, irritating air quotes, or the contraction of this myocardium. This isn't what causes your heart sounds. In fact, if you pause to think about it for just a minute, is there anybody around who understands the contraction of striated muscle fibers better than the people that are watching me right now, who just survived 223? I think not. So you understand how muscle cells work. And what did your lecture professors tell you last semester about the sounds made by these sarcomeres and myofibrils and muscle fibers? Probably a great big nothing, because they don't make any sound. Everybody watch. I'll demonstrate. Skeletal muscle fibers. Listen very closely. And I have the microphone turned on. I didn't go out and spend all this money on fancy microphone equipment, so you don't have to listen to the ventilation quite so much for no reason at all. So listen very closely to the sound of skeletal muscle fibers as they contract. Oh, that wasn't loud enough? Allow me to double it up. I'll even do the crab like the bodybuilders. Oh. What sounds did the muscle cells make? They don't. They don't make any sounds. What you hear is actually the closing of the heart valves. First, the 2 atrioventricular valves, the tricuspid and mitral, when they snap shut, that produces a sound that you hear. That's the first of the Korsakoff sounds. So as blood is forced from an atrium down to a ventricle, the valves open and then they snap shut. That's a noise. The second sound you hear is the closing of the semilunar valves, pulmonary semilunar valve and aortic semilunar valve. When they close, after the blood is ejected from the ventricles, that's the second sound you hear. Many people like to say lub-dub, lub-dub. I don't really like to say that. I prefer to say, and think about this when you're in your lectures, I prefer to say down, out, down, out. So the two atria contract, forcing the blood down through the tricuspid and mitral valves, and then the two ventricles contract, forcing the blood out through the semilunar valves. So instead of lub-dub, or lubbed-up with the P, lubbed-up, some people say, I always think to myself, down, out, down, out, down, out, like that. So maybe that'll help you out in lecture a little bit. So the cause of the heart sounds is the closing of the valves. How do you listen to them? Well, you do that using a stethoscope. And I brought two in here to show you. Actually, one was already here. I pulled this out of one of my lab drawers. This is the student stethoscope, aka a piece of crap. And then I brought my own stethoscope down here from my office. I have one up there. Very nice, solid, heavy mast head or bell on this thing. This is a good microscope, a good stethoscope for you to listen to heart sounds with. The other thing that people wonder about is, how do you actually wear this thing? So I want you to look closely at these two stethoscopes, and I want you to see how the earpieces meet at an angle. They typically do that. See that? They meet at an angle. And what you want to do is set your stethoscope up so that the angle is pointing forward, or that it points the same way your nose does. It would be just like the bridge of your nose here, like this, so that the two earpieces are pointing forward into your ear canals. That tends to give you the best sound. So I line it up with my nose so it makes a peak just like my nose does, and then I put these two things in my ear canals. And then, of course, I can just drape it off my neck and walk around and be, all clinically cool, like some of you people will be. Maybe if I was wearing scrubs, it'd even be better. And, I'm just cool medico, like that. But that's how you wear it. So I'm going to put it on, and I've got this really fancy mic, so let's maybe see if you can hear some heart sounds. I'm going to put my fingers on either side of the diaphragm on the stethoscope, and I'm going to press, like I mean it, right at about the fifth intercostal space, a little to the left, like so. So listen closely, everyone. Oh, there it is. Sounds good, doesn't it? Nice heart sound. Okay, nothing wrong with that. He's alive. And, for all of you in my lecture, he actually has one. Yes, he does. Okay, now, what if, I know, what did you just hear? What did you just hear? That wasn't actually my heart through this stethoscope because the microphone can't pick that up. That was just a sound I was making in my throat, like that. In fact, I know it wasn't my actual heart that you heard because my heart does not sound normal. Whereas your heart typically down, out, down, out, down, out. Mine has actually two extra noises. Mine would be a little more like this. I have two heart murmurs, believe it or not. What's a heart murmur? A heart murmur is an extra sound produced by some type of abnormality or defect to one of these four valves. So what I have, I know, trust me, when my mitral valve, the left AV valve, closes, it flutters a little bit. I have a mitral valve prolapse. So instead of my heart makes this noise, like that. I also have a little stenosis of my pulmonary valve narrowing, so it makes a slightly abnormal sound. Trust me, I have been a quiz and practical question for many, many people. Student nurses, student doctors, EMTs, sonographers, you name it. One of the more common things people come back and find me when we actually have in-person classes, one of the most common reasons they come back and find me is to ask if they can listen to my heart murmur to see if they can detect one or the other or both of them for practice. The only thing that might be more common 224 students is when students come back to find me to ask me to re-explain the whole respiratory metabolic acidosis alkalosis thing that you're getting later on this semester. So, the core cough sounds are caused by the closing of the heart's valves, AV valves, tricuspid and mitral first, semilunar valves, pulmonary aortic are the second sound. Remember, I like to think down, out, and extra sounds caused by a defect to a valve. So you're supposed to know what the sounds are called, what causes them, and the cause of a murmur. Always go with that list of things to know. So continuing in our cardiovascular world, the next thing we have is the electrocardiogram, ECG. You'll notice in your manual it's also referred to as EKG. Don't say EKG, that will make people think you're as old as I am. Say ECG, the modern acronym, electrocardiogram. What you see in front of me is a simple laptop computer and what's called a power lab box that actually generates our electrocardiogram. Now you probably can't see, I'll just tell you, I have a foam rubber pad and an actual rubber pad under this box to electrically isolate it. It's sort of tough once in a while in this room with all the electrical interference we have to get a good ECG, but I'll give it the old college try, everyone, and see what we can produce. Your job for lab number 1 is to know the parts of an ECG waveform, what causes each, and how to calculate heart rate from an ECG. The electrocardiogram is a very passive recording of the heart's electrical activity. Perhaps back in your first semester course, you saw myographs, recordings of what muscles do. This is an electrical myograph, if you will, of what the myocardium does. So I want you to follow along in your lab manual, paying particular attention to the questions you have on page 9 that we'll go over in just a moment. And don't panic if you cannot see the screen here, because I will reposition the camera so it's hopefully very visible to you. So the first thing that you always want someone to do is take off any jewelry or electrical appliances, watches and so on, from their wrists and their hands. You also prefer to have them in an electrically isolated room. Maybe some of you working in the field or as a patient have seen that ECGs are sometimes recorded in a room with the lights low, not a lot of electrical appliances. Sometimes in this particular lab room, the electrical activity in our ceiling, in the walls, can disrupt our electrocardiogram. That's why I try to isolate the box as much as I possibly can. We're going to use a simple 3-lead ECG, red, black, and green for ground. And of course, these clothespin leads, they're called, are very easy to attach. So we don't get as highly accurate an ECG as you might at a hospital or a medical facility, but oftentimes we can get one that's good enough. I'm a big believer in the use of a good amount of electrode gel to produce, let's hope, a better signal from our ECG. And our clothespin leads are labeled. So black on the left wrist, see the receiving plate there, little metal plate. So I put this one on the left wrist. Red goes on the right wrist. Smear some gel around on my plate. And the green is the ground, which typically goes on the ankle or up higher on the forearm of my patient, in this case myself. And then my job as the patient is really to just sit still and not try to move around very much. So I'm going to go ahead and click the start button here to start producing an electrocardiogram. And I will just sit as calmly as I can and see if we can get a fairly decent trace here. And I notice one of my wires is loose. So let me put it back in. This is what happens in the real world, is sometimes you have a loose wire. So now I'll just try to relax and see if we can get a good run. Okay, that's perfectly good. Now let me reposition the camera and ungel myself so you can see what this thing actually looks like. Okay, everyone, what we have in front of you right here is a fairly standard and not bad electrocardiogram or ECG trace. So you can see the waveforms running right here, and this is just the one I recorded here just a moment ago. So let me zoom in a little more on just a couple of these waveforms. So let's look, you and I, at this one right here. So follow along with me and the diagram that's in your lab manual. I can see right here the P wave. This tall spike, very narrow, is the QRS complex, and this hill right here is called the T wave. I can see the same thing in the next waveform over. Everybody look, P wave right there, QRS wave or QRS complex right there, and T wave right there. P, QRS, T. When people count these things, typically what they are counting is the QRS spikes because they're very easy to identify. Now I mentioned before this is a passive electrical recording of what my heart was doing. The P wave right here represents atrial depolarization. And you have to say it that way. Atrial depolarization. The QRS here and here represents the depolarization of the ventricles. And the T wave represents the repolarization of the ventricles. So for a lab quiz, let's be very straightforward. If I say what happens right here at the P wave, you must say, here's my wagging finger, you must say atrial depolarization, the electrical activity of the heart. If I point at a QRS complex, like right here, you must say ventricular depolarization. And if I point at the T wave, you must say ventricular repolarization. You cannot use terms like relax and contract because this is an electrical recording. You have to tell me about the electrical events of the heart at these various points in the waveform. So each one of these trio of waveforms, PQRST, PQRST, represents a cardiac cycle, at least in the electrical sense. You will notice there's no waveform showing me atrial repolarization. I'll leave that for your lecture instructors to describe for you, why that is. But for lab, what's this called? P wave. What's this called? QRS. What's this called? T wave. Now, yes, you notice my T waves are very tall. That's not that unusual. So this is a perfectly normal looking ECG, and not a bad one, given all the electrical noise that's in this room. The other thing you have to be able to do with an ECG is to be able to calculate a heart rate using one like you see in lab. Notice at the bottom I have a timeline that ticks the time by. So what I'll do using the scroll bar here, so I can see 1 minute even. So I could calculate a person's heart rate by taking a known amount of time. Let's say I start at 45 seconds right here, and I count how many QRSs there are in 15 seconds. So I'm going to do that, starting at 45. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 in 15 seconds, which would make my heart rate exactly 60 beats a minute. No, I didn't plan that. Let's double check and make sure I counted correctly, because I had a little stutter in there. Starting at 45 seconds, 1, 2, 3, 4, 5, 6, 7, 8, 9 to this one right past 53. Ten, 11, 12, 13, 14, 15. So 15 cycles, or QRSs, in 15 seconds. So I would take whatever number I got in my 15-second window, multiply by 4, and that tells me how many beats I would have in 60 seconds, or 1 minute. In my case, my heart rate is 60 right now, today. 60 beats per minute. Count the QRSs in a 15-second window and multiply by 4. If I had a 30-second window, I would count all the QRSs in 30 seconds and multiply by 2. Easy way to calculate a heart rate using an ECG. And in the practice section, I'll give you a couple practice questions so that you can do this.